Good morning. My name is Ann Brazo. I'm the founder and CEO of MPN Advocacy and Education International. Yesterday, we hosted a clinical trial update webinar for patients, and it was very well attended. And today it's for physicians. Usually, the physicians, a handful will watch today, and then others will come back on our website or on our YouTube channel and watch the recordings. Because they do want to know this information, and it's important that they know. We will begin this morning with Dr. Evan Bronstein, who's the senior medical director at Insight, and then move on to our other partners, and you will hear from several. And it was fascinating yesterday, very informative, and I'm sure it will be the same today. So, Dr. Bronstein, I will turn it over to you.
Great, thank you so much, Anne. And I will go ahead and share my screen. If you could just confirm that you can see my slides.
I can. Thank you.
Excellent. All right, so then I'll go ahead and get started. So as Anne mentioned, my name is Evan Bronstein. I'm a medical director at Insight, and it is really my pleasure to be able to review the clinical programs that Insight has ongoing in MPNs. Make sure I can advance. Okay. So this is an obligatory disclosure slide that I will just pause for a few seconds to allow everyone to read it. Okay, and now we will get into the interesting content. So Insight has multiple clinical development programs that I'm going to review today, and they are shown here on the screen. Of course, hopefully most of the audience is familiar with our JAK inhibitor ruxolitinib that is currently FDA approved for use in MPNs. I'm not going to review that compound today. I'm only have it here to mention that our current clinical programs do use ruxolitinib in combination with the investigational therapy. I am going to review 4 other compounds that are shown here. We have a BET inhibitor, an ALK2 inhibitor that are ongoing in clinical trials. And then we have 2 newer concepts, our mutant CALR program and our JAK2 V617F specific inhibitor that I will also review today. So I'm going to start with a brief review of our BET inhibitor. This trial is our LIMBER 103 trial. BET proteins are epigenetic readers, epigenetic modifiers that are involved in regulating transcription of multiple genes. And there is significant both preclinical and clinical evidence that they are active in myelofibrosis as well as other MPNs. And one of the proteins that they're known to regulate is NF-kappa B, and modulation of that protein is thought to be part of some of the efficacy that is seen in MPNs. And so Insight is investigating BET inhibition in the treatment of MPNs. We have an ongoing phase 1 clinical study to investigate BET inhibition. We are studying this compound both as monotherapy as well as in combination with ruxolitinib. And you can see the study schema shown here. Currently, this program is, as I mentioned, this program is ongoing and is in the dose expansion phase. And so I'll go a little bit more detail into these 2 study arms. So our monotherapy program is being studied in relapsed refractory myelofibrosis as well as MDS overlap and ET as well. This program has advanced from dose escalation into dose expansion. So we have identified doses to use in expansion, and we have seen that one of the main activity of this drug is to reduce platelet counts. And so we are studying this drug in dose expansion in 2 doses, and patients are stratified based on their platelet count. So lower doses being studied in patients with lower platelet counts, while a higher dose is being studied in patients with higher platelet counts. And you can see we are studying this drug in monotherapy in MF, relapse refractory MF, as well as relapse refractory ET. We have two ongoing treatment arms, one in MF and one in ET, to study this compound in monotherapy. And this trial is open and ongoing. We are also studying our BET inhibitor in combination with ruxolitinib. The combination arm remains in dose escalation at this point, and we are studying this compound in myelofibrosis that has a suboptimal response to ruxolitinib. As we move forward with this compound and identify doses for expansion, you can see that the study schema is very similar to monotherapy. We will plan to identify two doses, one to study in patients with lower platelet counts and another to study in patients with higher platelet counts. And we plan to study this in MF, as well as accelerated phase MF, so patients with higher blast counts, and in a JAK-naive cohort, patients that have not been treated previously with JAK inhibitors. So that, again, this trial is also ongoing, and as I mentioned, currently is in dose escalation, and we hope to proceed to dose expansion very soon. The second study that I'm going to review is our ALK inhibitor called zilurgisertib. We are studying this molecule also in monotherapy and in combination with ruxolitinib. And so the indication for ALK inhibition is a little different than our BET inhibitor. ALK inhibition is really designed to target patients with anemia, and that is because there is preclinical and clinical evidence that ALK inhibition can reduce hepcidin levels. And we know that the majority of patients with myelofibrosis have increased hepcidin, and we think that that contributes to the anemia that occurs in myelofibrosis. And we know that anemia is a significant burden in patients with myelofibrosis. And so ALK inhibition is being studied in MF patients, myelofibrosis patients with anemia. This is the study schema. This study is ongoing and is in dose escalation. Similar to our BET inhibitor trials, we have multiple arms. We're studying zilurgisertib in monotherapy, as well as in combination with ruxolitinib. And the combination arm has both a patients with anemia with relapse or suboptimal response to ruxolitinib, and a treatment-naive arm for patients who have not been previously treated with a JAK inhibitor. And this study is ongoing and is currently in dose escalation. And again, we hope to identify safe dose for dose expansion relatively soon and expand these trials as well. And then the next two trials the next two compounds that I'm going to review are newer compounds. I'm first going to talk about our mutant CALR selective monoclonal antibody, and then talk about our selective JAK2 V617F inhibitor. Our mutant CALR antibody targets the mutant CALR protein, and I think most of the audience is aware, mutations in the CALR protein are one of the drivers of MPN and occur in approximately 30% of patients with myelofibrosis and ET. The wild-type CALR protein normally resides in the endoplasmic reticulum. However, when it's mutated, it traffics through the endoplasmic reticulum, binds to the TPO receptor, and goes to the cell surface. And that complex of TPO receptor and mutant CALR causes cytokine-independent activation of that receptor and JAK-STAT pathway activation, and we believe that's what leads to the disease. And our antibody is designed to specifically target the mutant CALR protein and inhibit this cytokine-independent activation, and thus it represents a tumor-specific inhibitor in MPN, and so it's one of the first of its kind to be able to do that. This is the mutant CALR protein, and our antibody targets the mutant-specific C terminus, as I mentioned. So the C terminus is the part of the mutant CALR protein that changes in the mutant. And so our antibody targets specifically that portion of the protein. It's a fully human IgG1 and Fc silent antibody. And our study schema is shown here. So this study is open to enrollment. It is currently in dose escalation. We have two arms. We are studying it both in myelofibrosis and in ET. Currently, it's open for patients who have been previously treated with JAK inhibitor for MF cohort, and for ET patients who are resistant or intolerant to at least one cytoreductive therapy. We are currently, as I mentioned, in dose escalation, and this study is ongoing. And then the last molecule I'm going to talk about is our JAK2 V617F inhibitor. This protocol or this trial is not open yet, but hopefully will be in the near future. And so here I'm just presenting the mechanism of our inhibitor. We have developed a oral molecule that is specific and preferentially binds to the JAK2 V617F mutant protein. The V617F mutation, which is the most common mutation that occurs in MPNs, is in the pseudokinase domain of the JAK2 protein. And the normal function of the pseudokinase domain is to help stabilize the JAK2 protein in an inactive state. And when the V617F mutation occurs, the JAK2 protein is preferentially in the active state and can dimerize and lead to cytokine-independent activation. And the molecule that we've developed specifically binds to the V617F mutated protein and prevents this dimerization and cytokine-dependent activation. And so we hope, similar to the mutant CALR antibody, that this molecule will be a tumor-specific inhibitor and target only the tumor cells, leading to specific inhibition in patients with myelofibrosis. And so, as I mentioned, this trial is not yet open, but hopefully will be in the near future. And that concludes the talk. And I hope you have enjoyed and learned a little bit about some of the programs that Insight has ongoing. Thank you very much, Anne.
Thank you, Dr. Bronstein. And that was very informative. It's just fascinating how much is happening still in the MPN space. And I'm really curious about the CALR target. That's very interesting. Thank you so much.
Right. Thank you.
And you have a good day, and we will move on. Stay with us if you'd like. We're going to move on to SOBI and Olivia Cholak, who is the medical science liaison. Olivia.
Great. Thank you so much, Dr. Bronstein. Thank you for that. That was fascinating. You guys are doing really incredible things. Let me get my presentation up real quick. Okay, can you see this?
Yes.
Okay, I think we're good
Or
to
no,
go.
I can't
Great.
see it. I cannot see it.
You cannot? Hold on one sec. Okay, let me get it back up again. Let's see. Okay, can you see this?
Yes, but it needs to be the full
terrific.
screen.
Yep.
You need the full screen.
Okay.
There you go.
Great. Thank you so much. So hi, everybody. And again, thank you so much for having us join you today. This is a really fantastic panel to be a part of. My name is Olivia Cholak, and I'm a medical science liaison for SOBI in New York City. And today I'm here to talk to you about pacritinib, brand name Vonjo, and its use in thrombocytopenia and myelofibrosis. And then give you an update just on our most recent research efforts. As I said to the patients on the call yesterday, my background is in nursing. I worked in the MPN leukemia clinic at Mount Sinai for many years, and it was really a transformative experience for me. So, it's really, I have a really personal connection, to MPN and myelofibrosis patients. And again, just really glad to be able to be on the medical affairs side of things these days and helping patients in a new way. So the presentation that I have for you today is actually relatively light on data. But for those of you who might be unfamiliar with Vonjo or with Sobi, I'm really just looking to give a high-level overview of who we are as a company, just go over the unmet need for myelofibrosis patients with severe thrombocytopenia, and then go over Pacifica, which is our ongoing confirmatory phase 3 trial. So Vonjo was developed by a company called CTI, which was acquired by Sobi 1 year ago. Sobi is a global leader in rare disease, and now that the two companies have merged, Sobi is really showing itself to be a leader in the hematology space as well. And really, Vonjo is really the crown jewel of Sobi's portfolio. So, what sets Vonjo apart from the other myelofibrosis therapies is that it's the only JAK inhibitor that is specifically for severely thrombocytopenic patients. So, those are patients with platelets of less than 50,000. And as we're going to kind of come back to time and time again in these slides, this population really represents a significant clinical unmet need before the approval of Vonjo. We all know that myelofibrosis is a very rare disease, affecting less than 80,000 patients worldwide. For the purpose of this talk, I really want to highlight the severely thrombocytopenic population. So again, those patients that have platelets of less than 50,000, and that's about a third of our patients. And we know that low platelet count is really a poor prognostic indicator in myelofibrosis. So these patients have a mean overall survival of, just about 15 months. And that's compared with 57 months in patients that have baseline platelets of over 100,000. So really, we see quite a considerable difference just based on platelet count alone when we look at survival. We know that patients that have lower platelets also have a higher symptom burden. They feel worse. They're definitely plagued by the symptoms that they experience, the fatigue, the night sweats, the bone pain. This is really a very sick group of patients, and unfortunately, there aren't, as I've said, many options for these patients. To begin with, before PERSIST-2, which was the phase 3 trial that led to Vonjo approval, no study looking at JAK inhibitors had even looked at patients with platelets of less than 100,000. And then, of course, that we know that myelofibrosis treatments themselves can be myelosuppressive. So, when putting a patient with severe thrombocytopenia on a JAK inhibitor, these patients are oftentimes they're subtherapeutically dosed. They're not, getting any optimal effect as far as spleen and symptoms are concerned. And they've really been left with very few options. But this is where Vonjo comes in. And Vonjo is the first FDA-approved medication for severe thrombocytopenia in myelofibrosis. And it shows a spleen and a symptom benefit in patients that have platelets of less than 50,000. We also have safety data for platelets of less than 100,000, and I'll get in a few slides into our NCCN guidelines. In PERSIST-2, which was that phase 3 trial, 22% of Vonjo patients had a spleen volume reduction of at least 35%, compared to 3% of best available therapy. The best available therapy arm was primarily low-dose ruxolitinib. 32% of patients had at least a 50% reduction in their total symptom score, compared to 14% of best available therapy. And really of note is that these patients, who could not necessarily tolerate full dose on another JAK inhibitor are able to tolerate full dose of Vonjo, and they can really reap all the spleen and symptom benefits that Vonjo can offer. Now, what really sets Vonjo apart and what makes it unique is the unique mechanism of action and the pathways that it not only inhibits, but the pathways that it spares as well. So Vonjo is the only JAK inhibitor to spare the JAK1 pathway. And it's thought that this is why Vonjo preserves hematopoiesis and preserves immune response. So we don't have any warning for infection with Vonjo. Like all the other JAK inhibitors, it does target JAK2 pathway to address spleen size and symptoms that we see in myelofibrosis. And on top of that, we have the added benefit of also inhibiting the IRAK1 pathway. That's going to dampen NF-kappa B transcription and that pro-inflammatory cytokine production downstream to even further address those inflammatory symptoms that we see in MF. And, you know, Dr. Bronstein had just talked about hepcidin. Vonjo also does inhibit the ACVR1 pathway. What that does is it inhibits hepcidin transcription. So in turn, that's going to increase available iron stores in the liver for hematopoiesis, and that may be why we see an anemia improvement with Vonjo as well. I just want to highlight our inclusion in the NCCN guidelines. Here, looking at the treatment algorithm for high-risk myelofibrosis, we see that it is the preferred regimen, category 1, for patients that have platelets of less than 50,000 who are not transplant candidates. And it is also included as an option for patients with platelets of greater than 50,000 as 2B. So it can be given any line agnostic of platelet count. We're also included in the treatment algorithm for myelofibrosis-associated anemia. And remembering back to that MOA slide, the implication of ACVR1 inhibition. We have some really exciting anemia data that I know if you're looking for some more details, if you can get in touch with me or any other MSLs here at SOBI, we'd be happy to share the data with you. But just really briefly to go over this, for patients who are anemia with spleen symptoms that are controlled, PAC is listed as being useful in certain circumstances. For anemic patients whose anemia, spleen, and symptom are not controlled, PAC is listed as a recommended regimen. And then it's category 2B for any anemic patients without symptoms. And now getting into our ongoing trial, Pacifica, here's just a rundown of, a timeline of events leading to Vonjo approval. So Vonjo was approved by the FDA in 2022, and it got accelerated approval for patients with platelets of less than 50,000 based on spleen volume response that was seen in PERSIST-2. The accelerated approval is based and contingent upon the completion of the Pacifica trial, which started enrolling in 2019. Now, Pacifica is still ongoing, but as of 2022, when Vonjo was approved in the United States, all U.S. sites for Pacifica closed. So now it is only an ex-U.S. study. So it's a global study for Pacifica. Here's the study design for Pacifica. Really, the key points here are that this is looking at MF patients with intermediate or high-risk disease with baseline platelets of less than 50,000. And these are patients that are JAK inhibitor naive, or they've had a JAK inhibitor for a limited duration previously, so less than 90 days. It's looking at 399 patients that are going to be randomized 2-to-1 to receive either pacritinib or physician's choice. So physician's choice includes low-dose Rux, so 5 milligrams daily or twice daily, hydroxyurea, danazol, or corticosteroids. Then, of course, co-primary endpoints, like all JAK inhibitor studies, are spleen volume reduction and TSS reduction. And then for key secondary endpoints, we're looking at overall safety, patient global impression of change score, and then, of course, safety. This is just reviewing those objectives that we just went over. And then here is just a map looking at our study sites throughout the world. So we've got approximately 100 clinical sites worldwide. We currently have just over half of the study enrolled. So a little over 200 patients are enrolled in Pacifica right now. And we're on Amendment 10 of the protocol, which was put in place to allow for increased eligibility to allow more patients to enroll in Pacifica. Here, I'll just give you a moment to look at our indications and our safety information. And that brings us to the end of this one. Again, I really appreciate you listening. Please don't hesitate to reach out with any questions and enjoy the rest of all the presentations today.
Thank you, Olivia. Very informative. Appreciate it. And moving right along.
Thank you.
Oh, you're very welcome. Moving along, we will hear from Karyopharm Therapeutics and Dr. Raman Garsha, who's the Senior Medical Director of Clinical Development. Hello again, and thank you for joining us again.
Hello again. Thank you, Anne, and thank you everyone for your time. Let me just and I'll just adjust my display setting here. Okay, how does that look?
That looks good.
Thank you. So as Anne mentioned, my name is Raman Gartra. I'm the Senior Medical Director here at Carrier Farm. And today, I'll be talking through our mechanism of action, our phase 1 to phase 3 trial, which we've now rebranded, and we have a brand new study name, which is Sentry. And then also discuss the plan for our phase 2 trial, now called Sentry 2. So there's a disclaimer slide here. Okay, who are we? So Carrier Farm is an innovation-driven pharmaceutical company, and we focus on the development and commercialization of these novel first-in-class agents. So, I'll be talking about selinexor today, which targets nuclear export and related pathways. The company Carrier Farm, we have an experience of 10 plus years in research and development, and we're headquartered in Newton, Massachusetts. So I mentioned our phase 1 to phase 3 study, Sentry, and our phase 1 study was a dose-finding study, obviously, with enrollment complete. Our phase 3 Sentry study is now open for enrollment in a combination of selinexor with ruxolitinib in JAK-naive patients in myelofibrosis. And our phase 2 study, Sentry 2, is also open for enrollment, and I'll be discussing these studies in the presentation. So just to touch on, some of the well-described unmet needs in myelofibrosis. And, generally there's a need for therapies beyond standard of care here, and, improving on SVR35 and TSS50 responses that we see in standard of care. Also, there's a suggestion standard of care has a limited ability to disease modify, which is a really hot topic, obviously, in order to help us see these improvements in long-term survival for MF patients. Also, management of cytopenic MF patients is something that we need to address. Very often there's limited treatment options, and standard of care may be used in a suboptimal dosage for these varying degrees of thrombocytopenia, for example. Okay, so what you can see here is a slide on our mechanism of action. So selinexor is a first-in-class agent. We have data. You can see the references at the bottom of the slide that referred to preclinical and human cell line data. And selinexor is an XPO1 inhibitor, basically inhibiting the export of cargo proteins from inside the nucleus. And you can see with the circle of the cell there, so that's a cell, and the blue is the nucleus. And we've color-coded this just to, in a general way, indicate the various mechanism pathways that, our evidence suggests we see for selinexor. So, as well as selinexor acting on the JAK-STAT pathway inhibition, we also see p53-driven cell death, cell cycle arrest, and NF-kappa B pathway inhibition, which may have an action on reducing cytokine production. What you can see here is the phase 1 plan for Sentry, formerly known as Export MF034. And obviously phase 1 was a dose-finding study where 24 patients were studied at 2 different dose levels, the 40 milligram and the 60 milligram dose levels. And in fact, it's the 60 milligram level that was chosen as the recommended dose. Just to go over the demographics very briefly, and please do refer back. I want to touch on the important parts of each slide. So in the 60 milligram group, as you'd expect, the majority were primary MF, JAK2 driver mutation driven. In terms of DIPS, intermediate 2 was the highest proportion, with equal high risk and intermediate 1 proportions in the 60 milligram group. What you can see here is the Sentry phase 1 safety summary. And, just to note, hematological AEs that you would expect to see in this group of patients were manageable by appropriate dose modifications. We did see nausea as a treatment emergent effect, but this was transient in nature and manageable with prophylactic antiemetics. We also saw a 2.5 kilogram median weight gain comparing baseline to week 24 for the 60 milligram group. And, notably, in this group, we found hemoglobin and platelet levels were generally stable when comparing baseline to week 24. So, we're very excited about the efficacy that was seen in phase 1. So again, this is selinexor 60 milligrams in combination with ruxolitinib. And on the left-hand side of the screen, you can see SVR35. And on the right-hand side of the screen is TSS50. So, in both cases, we see early responses by week 12. And in fact, if you go to the intent to treat population, by week 24, 79% of patients achieved SVR35, and 58% of patients achieved TSS50 by week 24. What you can see from the figures just at the bottom of the slide there is, in fact, all patients in the efficacy evaluable population achieved an SVR35 at any time. So, we often refer to the week 24 time point, but in fact, achieving a response at any time in treatment is also important. Responses were found to be durable. There's 100%, these are flat line curves here, as you will. So, there was 100% probability of maintaining SVR with a median follow time of 32 weeks and 100% probability of maintaining TSS50 with a median follow-up time of 51 weeks. So I spoke briefly about disease modification earlier, and there were suggestions from our phase 1 data that we do see some of this, and this is to be further explored in our phase 3 trial, which is now enrolling. On the left-hand side of the screen, you can see in both dose groups, 60 milligrams and 40 milligrams, a reduction in cytokines seen early at week 4 was associated with some level of spleen volume reduction at week 24. On the right-hand side of the slide, you can see, again, in both dose groups, 60 and 40 milligrams, there was reduced allele burden regardless of the driver gene mutation that were observed in 13 of our evaluable patients, some of whom also had high molecular risk disease. So what we have here is the study plan for Sentry phase 3. So we're aiming for enrolling 306 patients globally. Again, JAK-naive patients with myelofibrosis. This is a double-blind trial with a 2-to-1 randomization. The two arms being 60 milligrams, and this is dosed orally, PO, once a week, QW for selinexor, in combination with ruxolitinib BID. And given the platelet count inclusion for this trial of 100 plus, we're expecting 15 and 20 milligram ruxolitinib doses to be used. The co-primary endpoints are SVR35 and TSS50 at week 24. You can see the eligibility criteria here on the screen, and they're very similar to other MF trials, phase 3s that are running. DIPS, just notably DIPS intermediate 1, intermediate 2, or high-risk patients, and primary MF or secondary MF. I'm going to finish off the talk here describing Sentry 2, which we're very excited about, which is our phase 2 trial, which is now enrolling. So, I want to touch on some of the rationale for this trial. So in our phase 3 trial, Sentry, phase 1 trial, sorry, Sentry, that I just described, we had a group of patients that were on selinexor 60 milligrams, but also on a suboptimal dose of ruxolitinib, i.e. 5 milligrams BD or less. So even where this very low dose of ruxolitinib was used in Sentry phase 1 in combination with selinexor, we still saw SVR35 and TSS50 responses. So that may suggest that there's some selinexor monotherapy activity here, which leads us to Sentry 2, our phase 2 study. And you can see the study plan here, and it's quite a novel design. So Sentry 2 will be using selinexor 60 milligrams monotherapy upfront for JAK-naive myelofibrosis patients, but importantly, in a moderately thrombocytopenic population. So this is an area of unmet need in myelofibrosis, which we are trying to address. So the platelet count inclusion for this study is 50 to less than 100 times 10 to the power 9 per liter. There's two dosage arms, 60 milligrams and 40 milligrams. And the other really interesting design feature for this study is at weeks 12 and 24, if certain SVRs are not met, it's at investigator discretion to add on one of 3 JAKIs, so ruxolitinib, pacritinib, or momelotinib, obviously depending on the platelet count and the hemoglobin count here. That's a really nice option for patients to have. And we've had some very good feedback from KOLs on this study design. So I'm going to stop there and, thank you all very much for your time.
Oops. I keep coming and going here. Thank
Yeah.
you very, very much. Again, just as yesterday was fascinating, it remains so today. Thank you very much for joining us today. And we
Thank
will
you,
move
Anne.
on to Dr. Efode Ajari, who is the Executive Director, the Medical Executive Director of Clinical Development at Protagonist Therapeutics. Hello again.
Hello.
Thank you for joining us again today.
Thank you very much for having us. Hello, everybody. Efode Ajari here today again, part of the clinical development group within Protagonist. For those of you that do not know enough about Protagonist, we are a biopharmaceutical company located in the South San Francisco Bay Area. We are a truly strong biotech company because our platform has very interesting assets, and the primary one is based on our proprietary peptide technology with rusfertide or PTG 300 as our lead investigational drug candidate. Rusfertide is a synthetic mimetic of the naturally occurring hormone hepcidin. And as you all know, I'm sure, hepcidin regulates iron metabolism and also controls the absorption, the storage, and distribution of iron in the body. Just as a way of setting the stage for our discussion of the disease, polycythemia vera, I just wanted to remind us about a few things. It's a myeloproliferative neoplasm that is characterized by clonal erythrocytosis. You see elevated hematocrit levels as a hallmark of the disease. Quite a number of serious long-term diseases and comorbidities that we see associated, from increased thrombosis that would affect the heart and other organs. Those can arise. PV can happen at any age, but it's more common in those people aged between 50 and 70 years with a median survival of approximately 20 years. The treatment goal here is just to keep hematocrit at less than 45, hopefully, in order to minimize possible complications. Often the disease is suspected when you see elevated hemoglobin levels and hematocrit measurements. Diagnosis, half of them might come with hypertension, a third even with some palpable splenomegaly. And you have other features like fatigue, early satiety, and all of the things, they can reduce the quality of life for patients, and the symptoms may worsen over the course of the disease. Therapeutic phlebotomies are what people do as mainstay, before they even add other things. But it has limitations, from fluctuations in hematocrit values to even the resultant iron deficiency anemias that we see. And as we know, many patients that are treated with phlebotomy, they tend to report more severe symptoms. And over half of the patients and physicians alike report that these phlebotomies are time-consuming, burdensome, and they can really very much affect the overall quality of life of these patients as well as their productivity. Okay, we've set the stage, but it's also very important to mention that in addition to phlebotomy, aspirin, hydroxyurea, interferons, as well as ruxolitinib, are some of those current treatment approaches in PV, especially in the USA. There are quite a number of ongoing interventional phase 3 studies in PV, from the use of interferon to even including low-dose aspirin or hydroxyurea, even to combining direct oral anticoagulants with low-dose aspirin as well. To talk about hepcidin, it's central to the regulation of iron metabolism. Its effect on a cellular level involves binding ferroportin, which is the main iron export protein, and it can result in its internalization and degradation, which ultimately brings about some iron sequestration within those ferroportin expressing cells. So any aberrantly increased hepcidin would bring about some systemic iron deficiency, usually with iron-restricted erythropoiesis. Rusfertide is a potent hepcidin mimetic, so it copies or mimics this mechanism that addresses this imbalance of iron metabolism by targeting that ferroportin receptor in a very much similar manner. So just like hepcidin, it will induce endocytosis and proteolysis of ferroportin, thereby decreasing the delivery of iron to plasma. So it's really very interesting. So through that effect of restricting iron availability for erythropoiesis, we started learning that rusfertide may be an effective therapy for conditions that are characterized by excessive erythropoiesis, such as PV. And that's actually how we got to this point in our development journey. As you may have heard, we very recently got into a partnership agreement with Takeda to jointly develop rusfertide, which indicates good news for our patients. Rusfertide is also being investigated, quite a number of phase 2 and the phase 3 clinical trials that we are doing for PV. We have the ongoing verified phase 3 pivotal study for about 250 PV patients. We just completed our enrollment a few weeks ago. There is the revived phase 2 study for 70 participants with its additional 3-year open label extension. Now, the core part of this study has been completed. The open label extension is still ongoing. And, there's also another long-term extension study for this revive and for the patients that enables those patients from revive to roll over. Now, there was the Pacific phase 2 study, which was a proof of concept study also for those patients with very high hematocrit levels that we actually had individual titrations of rusfertide, and the study has been completed. It's also very important for us to quickly highlight here that rusfertide has orphan drug designation and fast track status for PV. Okay, I would use this opportunity to present our phase 2 revive trial. For this trial, prior to giving our first dose of rusfertide, all the patients were phlebotomized to hematocrit level of less than 45%, just to have a standardized starting hematocrit for all of them. Rusfertide doses were then given subcutaneously in addition to their prior standard therapies. Our clinical goal here was to maintain hematocrit at less than 45%. We performed quite a number of analyses. We looked at the analysis of response. We were assessing the reduction in phlebotomies, and we also used the validated myelofibrosis symptom assessment form as a tool to measure the symptomatic responses as reported by patients. And I will really very quickly highlight some of the results. But for the REVIVE trial, just to say a few things about the study design and the background, as I already mentioned, we're looking at the safety and efficacy of rusfertide in phlebotomy-dependent patients with PV that were treated with or without any concurrent cytoreductive therapy. Now, in the part one of this international trial, we enrolled 70 patients in a 28-week dose-finding assessment of rusfertide. The part two was the double-blind randomized withdrawal period, in which we assigned these PV patients in a 1-to-1 ratio to receive either rusfertide or placebo for up to 12 weeks. Now, the primary efficacy endpoint was a response defined as, hematocrit control, absence of phlebotomy, and then a completion of the trial during the 12-week treatment period within this part two. Of course, there was the third part, which was an open label extension for up to 3 years. We also assessed, using patient-reported outcomes, we were assessing the symptoms of these patients as they reported it, using the modified MPN symptom assessment form I mentioned previously. And we required all the patients to, of course, they'll have PV, but at least 3 therapeutic phlebotomies in the prior 28-week period prior to enrollment, with or without concurrent cytoreductive therapies. Then they were also phlebotomized prior to the study. And we'll be seeing some very interesting data very shortly. Okay, these are the key demographics and disease characteristics. As you see here, 58 of the 70 patients that enrolled into part one continued into the open label extension. And just know that over half of these were actually high risk. Over half of these, they were actually high risk. I would also like to use these swimmer plots to tell the story of how rusfertide decreased the frequency of phlebotomies with or without concurrent cytoreductive therapy. The red dots you see indicates phlebotomies, and you see across the board that in the 28-week period before the first dose of rusfertide was given, these patients had quite, a very high frequency of phlebotomies before they got the first treatment. Now, 32 of the 58 patients were treated prior with phlebotomy alone. 26 were receiving concurrent therapy with cytoreductive therapies, plus phlebotomy. Now, before they started treatment with phlebotomies, they all had an average of over 4 phlebotomies during this period. The mean maximum hematocrit in the participants were way over 50%, but this level fell to about 44% once rusfertide was added to their treatment. And as a result, almost none of these participants needed a phlebotomy during the first part of the study. And all these patients were essentially phlebotomy-free while they were on rusfertide treatment. Now, okay, we are looking at part two still, the blinded randomized withdrawal part of the study now. As we mentioned before, responders are defined as those subjects that did not receive any phlebotomies and they maintained hematocrit control without becoming phlebotomy eligible, and they completed 12 weeks of treatment. 62.9%, that's 18 out of 26, responded to rusfertide in our full analysis population. That is, if you exclude those that discontinued early and did not complete this part, as compared with 18.5 of those that received placebo. But even then, if you include them, you see that 60% of patients, that's 18 out of 30, responded to rusfertide in the randomized population, as compared to 17%. This result really clearly indicates an undeniably impressive point that rusfertide met the primary efficacy endpoint for response. Then just to add a few more thoughts, the study also showed that we were able to provide, using rusfertide, sustained and durable control of hematocrit, with this treatment resulting in a consistent maintenance of hematocrit at less than 45% through 2.5 years, and this is still ongoing for a number of the patients that we are still tracking. The patient-reported outcomes, they were assessed using the MPN SAF form, and between baseline and the end of part 1, rusfertide treatment was associated with a decrease across the board in individual symptom scores. For those patients with moderate or severe symptoms at baseline, so it was really very impressive. Based on the REVIVE study, we also reported very good long-term safety profile for rusfertide. The most common treatment-emergent adverse events were injection site reactions, which really were mostly localized, were mild to moderate in severity, and generally they decreased in incidence over time. Then they were all deemed to be histaminic in nature, and the use of any of the over-the-counter antihistamines were found to be helpful in these cases. Overall, about 77.1% of these treatment-emergent adverse events were moderate at most. About 21% were grade 3. There were no life-threatening or fatal TEAEs reported. And let's actually just note that the average duration of exposure was about 105 weeks. Overall, 14 patients, about 20%, experienced serious adverse events. There were 3 cases of basal cell carcinomas. There was 1 case each of atrial fibrillation, MI. We saw the case of genital dysplasia, and so on and so forth. We also had some long adenocarcinoma, a stage 1 malignant melanoma, then the AML and the squamous cell carcinoma, both seen in 2 placebo patients. Going on and on, as we are all aware, we've reported that these SAEs were observed to be consistent with the kinds of disease that we anticipate as comorbidities in the PV population. Most of the SAEs were assessed as being unrelated to rusfertide by the treating investigators. So just to enable us move forward, with potential registration, we designed a phase 3 study using the data that's really very impressive from what we have learned from the REVIVE study. And this is a 3-part pivotal study with a key inclusion criteria, which is to enroll patients that are at least 18 years old that meet the revised 2016 WHO criteria for the diagnosis of PV. Then they also, as we showed in the REVIVE study, required to have at least 3 phlebotomies due to inadequate hematocrit control in the prior 28 weeks. In the prior 28 weeks before they are enrolled, or at least 5 phlebotomies within the year leading to randomization. Now, for these subjects, once enrolled, they are initially randomized into the double-blind placebo-controlled period for 32 weeks. During this time, they are given either rusfertide or placebo as an add-on therapy to their ongoing treatment for PV, and randomization stratified by ongoing PV therapy. The second part is the open label treatment phase, during which everyone that completed the part 1A successfully would get rusfertide for an additional 20 weeks, which is from week 32 through week 52. And the third part, which we tag part 2, becomes a long-term extension for up to 104 weeks, during which all that completed the part 1B will continue to receive rusfertide. And the key endpoints are the proportion of those patients that get rusfertide achieving response, which is defined as the absence of phlebotomy eligibility, measured in the second half of part 1A between weeks 20 and 32, and also the mean number of phlebotomies within the double-blind treatment period. Just to mention, as I highlighted earlier, this pivotal study has already been completed. We actually just completed the enrollment, in the last couple of weeks. So we currently observing all the patients that have been enrolled as they are getting the treatment while on the study. These are really interesting times to think protagonist. I want to just conclude by, just highlighting that we've been able to show that rusfertide, number one, appears to be well tolerated in the REVIVE study, and we're actually seeing very rapid, sustained, and durable hematocrit control, as well as very positive improvements in symptom scores as reported by our patients. I also would want to just still mention that we continue to see results that are consistent with what we have learned from the REVIVE study in the current ongoing VERIFY study. We are really very pleased, and we'll continue to explore all the gains we would get from rusfertide. We'll keep updating you all as we learn more. I'll actually just stop here now. Thank you for this opportunity for us to talk about rusfertide, and I really look forward to collaborating with you all. On behalf of Protagonists, we thank you once more. Thank you.
Thank you very much. We appreciate it. Very informative. And now we're going to move on to Merck & Company Incorporated with Dr. Uzor Agbu, who is the Senior Medical Director there. And Dr. Agbu? I see your slides, but I don't see you or hear you. There you are.
There we go. Yeah, I remembered to share the slides, but not start
thank
the video.
you.
Yeah.
And so we will hear from you again today, and thank you very much for joining us.
Okay, again, thanks, Anne, for the invitation. I'm here today to talk to you a little bit about bomedemstat, an LSD1 inhibitor that is being developed by Merck. I'll start off with a discussion of LSD1, which, as we know, is an epigenetic enzyme that demethylates histone 3K4 and chromatin proteins that has a lot of, among other things, this is what it does. And we know it's a key regulator of hematopoietic differentiation. On the right-hand side there, you'll see sort of the expression of LSD1 mRNA across the cells all the way from hematopoietic stem cells, and it decreases all the way as you go down to mature cells. And LSD1 activity is important in the differentiation of megakaryocyte and erythroid progenitors. We also know that inhibition of LSD1 leads to expansion of the long-term hematopoietic stem cells without loss of self-renewal, and it leads to a loss of self-renewal in mutated mutation-bearing hematopoietic stem cells. We know that JAK2, CALR, and MPL are the mutations that primarily lead to a variety of different MPNs. Which one goes, it varies by patient, but those three mutations are the primary drivers of MPN development. So inhibition of LSD1 would lead to loss of self-renewal of those mutation-bearing hematopoietic stem cells. And that's important because that speaks to why we believe that bomedemstat would have activity across the range between ET, MF, and PV. Speaking a little bit more about bomedemstat, I've already said it's an irreversible LSD1 inhibitor. It's dosed once daily based on the PK and dose response, and it's also titrated based on the platelet count. So it goes up and down. We know patients have different amounts of sensitivity to LSD1. Some patients at lower doses have significant drops in their platelets. Others have a less significant. So a wide range of doses are required, which is why we need the titration. If we look at preclinical models, we see that they're treating with LSD1, an LSD1 inhibitor leads to a decrease in the malignant clonal population in bone marrow cells. It leads to a decrease in inflammatory cytokines, symptoms, spleen size, and bone marrow fibrosis. I'll show you a little schema on the next slide that talks about that a little bit more. But if you look on the right, these are the spleen and bone marrow from mouse. On the left hand, you'll see the inflammation and fibrosis in the spleen and bone marrow. After treatment, on the right-hand side, you see a significant reduction in that inflammation and bone marrow fibrosis. We also know there's no significant, doesn't cross the blood-brain barrier to any significant amount. And based on this, we think with its activity, both upstream and downstream, affecting not only the mutated stem cells, but also the cytokines and other things that lead on to all the symptoms that patients experience, that this could potentially have a disease-modifying effect. That will bear out as we continue studies. I will show you some data from the ET, our phase 2 ET trial, that gives us a little bit more confidence that that may be happening. So this is the digging a little bit deeper as to where it acts. Starting off with looking at the top sort of right-hand side, we know that the in this case, we're looking only at ET, but similar things would apply to the other MPNs. The activated megakaryocytes that are overproduced in essential thrombocythemia due to activation of the JAK-STAT pathway. LSD1 inhibition reduces the number of activated megakaryocytes, which leads to a reduction in the potential for thrombosis and bleeding. By reducing the cytokines, we see a reduction, a potential reduction in fatigue, anemia, and all the constitutional symptoms that patients experience. By reducing the production of growth factors, we have a reduction in reticulin and collagen deposition, which would lead to a decrease in bone marrow fibrosis, and that's reduced the need for extracellular hematopoiesis. One crucial and important part of it, I talked about a couple of times, is that LSD1 inhibition does affect the malignant stem cell population. It extinguishes self-renewal and reduces that stem cell population. Again, we hope this would potentially lead to disease-modifying effects. We are all not sure of what that would do over time as we continue to treat patients and as others develop data on variant allele frequency, and that we will see what impact that has. But it is a key feature of what LSD1 inhibition does in patients with myeloproliferative neoplasms. Based on that, at Merck, we have an extensive program cutting across both MF, ET, and PV. We have, in that first line there, you see the phase 2 studies that we have either ongoing or completed. So we have completed a monotherapy phase 2 study with MF that had some promising results. There is an ongoing collaboration in MF where we're combining it with ruxolitinib to see what that does. Some initial promising results were presented at ASH last year. I won't talk much more about that as we continue to develop more data. We'll come back and talk about that. In ET, that the phase 2 study has been presented at ASH. I will recap some of the results there and then talk to you about the phase 3 study that we launched last December, looking at second-line ET patients. And then I will show you the schema we have for our ongoing phase 2 PV study. That study is fully enrolled, but I'll show you what the schema is, and we hope to have results for that later this year. So this is the phase 2 ET study. We treated patients 18 years and older. They had to be an indication for cytoreductive therapy, so greater than 60 years or a history of thrombosis. You would expect those are mostly high-risk patients. And they also have an inadequate or a response or intolerant to at least one standard therapy. The platelet counts, hemoglobin levels are sort of what you would expect for an ET patient, ET study, where 450 and at least 100 grams. In this study, bomedemstat was given as a weight-based therapy. We have taken the data we have from this and converted it to a fixed starting dose. But in this study, it was weight-based. The phase 3 it has a fixed starting dose. And the endpoints we were looking at were safety and tolerability, and then a response as getting a platelet below 400,000 without any new thromboembolic events. The population that was actually recruited, the 73 patients, are largely what you would expect to see for a ET population that had received at least one prior therapy. The median age was about 66 years, which is expected. Between the mutation profile, JAK2, CALR, and MIPL accounted for about 90% of the mutations. There were also triple negative patients. We're at 9% there, which is also what you would expect. Most of these patients were coming off hydroxyurea. So you can see on the right-hand side there, 70% of the patients, their most recent prior therapy was hydroxyurea. Overall, 84% of the patients had been exposed, that had prior exposure to hydroxyurea. If we look at the safety profile, the conclusion is that it was generally safe. Most of the AEs were mild. As you can see, most of them were not grade 3 or 4. The most common AE, and the one we are still examining, is the dysgeusia. which in the case of patients taking bomedemstat results in an accentuation of taste. So sweet things tend to taste sweeter. It was mostly mild, it was limited, and we were able to largely control it with changing in dosing times and potentially lifestyle modifications. It is an interesting AE because, as I showed you, we have a wide program across the MPNs. We're not seeing the same level of dysgeusia across our MF and our PV patients. So we're continuing to examine, is there something specific to ET that is leading to this? And are there any other ways in which we can mitigate it? You also see there the thrombocytopenia. Obviously, that's a concern at 27%. This is largely due to it being the phase 2 study. It was weight-based dosing. We're trying to understand, how do we dose bomedemstat? We believe we have a handle on that now, and we should not see thrombocytopenia to this extent in our phase 3 study. There was also the GI symptoms and arthralgia, but again, all of these were largely mild symptoms and not really a cause for discontinuation. If we look a little bit at efficacy, focusing just on the platelet count here, patients had a mean platelet count starting at about 700. Coming off their other therapy and getting onto it, obviously we saw an increase in the platelets. And then as bomedemstat began to take activity, by about week 12, virtually all the patients had dropped down to the target platelet range of less than 400. You'll see 83% of patients overall received a durable hematologic response, defined as having at least 12 weeks of a platelet count less than 400. Of those that reached, of the 62 patients that reached treatment at least 24 weeks, 100% of those achieved a platelet count less than 400. And 98% of those did that in the absence of thromboembolic events. Going on to look, if we break down based on the type of mutation, we see that for those that stayed on treatment at 24 that reached 62 weeks, 100% of patients, regardless of mutation, JAK2, CALR, MPL, or triple negative, achieved the targeted platelet level. If we move on to talk a little bit about symptoms, you see for those patients that had a TSS greater than 20, 75% of them had a decrease in TSS, and 67% of them had a reduction of greater than or equal to 10 points. The figures here show you, are highlighting fatigue because that seems to be where we had the greatest impact. And you see they're moving from week 12 to 24, 36, and 48. We continue to see a reduction in fatigue when you look at the MPN SAF scale. Moving on to talk a little bit about changes in the variant allele frequency. We have noticed that there has been a reduction in variant allele frequency for patients taking bomedemstat. There you see 67% of patients had a reduction in their variant allele frequency while taking bomedemstat. What we have highlighted here is a few samples of a few patients. On the top right there, you see one patient who had a variant allele frequency of 46.3% at the start. That went down to 41.4 by day 169. And this carried out through, regardless of mutation. We had a few samples here of 3 JAK2 patients and 1 MPL patient that showed a decrease in their variant allele frequency. In the bottom right-hand corner, we see another interesting feature of bomedemstat treatment, where on day 1, that patient that you're looking at here had a significant proportion of their cells were homozygous for a JAK2 mutation. At day 169, you'll see that that portion of homozygosity decreased significantly, going with an increase in the heterozygous. It didn't change the wild type, but an increase in heterozygous. Expectation would be that going from homozygous to heterozygous would give you a less severe disease. Obviously, that will bear out as we continue to treat patients and correlate this with their symptoms in the course of their disease. So moving on to with that summary of what we had in the phase 2, moving on to the ongoing study, which is right now the lead study would be this phase 3 study in essential thrombocythemia, where we're treating patients who have had an inadequate response, or they're intolerant to hydroxyurea. The goal, it's a global study with the goal of enrolling 300 patients randomized 1-to-1 to either bomedemstat daily or to essentially any other available therapy that you would typically give a patient coming off of hydroxyurea. We have here anagrelide, busulfan, interferon, pegylated interferon, and ruxolitinib. And we're going to treat patients for a total of 1 year before we do our primary endpoint, but patients are able to go on to an extension phase, and depending on the results of the study, they may be able to switch from their best available therapy to bomedemstat at a later point. The primary endpoint for this is a durable clinical hematologic response, which is defined as having a platelet count less than 400,000 and absence of a white blood cell count elevation greater than 10 for at least 24 weeks during the 24 continuous weeks during this 52-week span that we're treating patients before we do the primary endpoint. So for those patients that had an elevated WBC count at baseline, we expect that to come down, and those that did not, we don't expect them to go up. And this is on top of that, there is also that there can be no thrombotic or major hemorrhagic events or progression to MF, MDS, or AML during that entire 52-week period. Here highlighting, this is the fixed starting dose for bomedemstat, 50 milligrams once daily, and it's titrated based on the platelet count. So as I said, the patient's sensitivity to bomedemstat differs. Some require higher doses and some require lower. In that phase 2 trial that I had talked about, we had a range of patients who were treated from about 30 milligrams all the way up to 100 milligrams to achieve their response. I'll also share with you, as I said, the phase 2 PV study, which is fully enrolled. We were targeting treating 20 patients there who had failed at least one standard cytoreductive therapy and had a platelet count greater than 250. We're treating those patients for a total of 36 weeks before we take a look at whether they achieved our primary endpoint, which is safety and tolerability, and a reduction of hematocrits to less than 45 without phlebotomy for at least 12 consecutive weeks during the 36-week period that we're treating these patients. Again, patients treated on this study are able to go on to receive additional doses of therapy as an extension. Here, given this is the first time we are treating PV patients, we did a little bit more conservative and started with a lower dose of bomedemstat, 40 milligrams per day. Again, still titrated based on the platelet count. As we come out of this study, we'll get a sense of what is a good starting dose for PV patients and to achieve a platelet count less than 45, hematocrit less than 45. And I'll return back to this slide just to, again, show across the different MPNs between MF, ET, and PV. We have a number of trials ongoing. We have our first phase 3 launch. We expect more to come in the future. And I'll highlight there on the bottom right-hand side there that we do have an open study with the bomedemstat extension study. You'll notice all of these studies we have here treating patients for about a year, a little bit more than a year, depending on how long it takes recruitment. But we do want to understand, what does bomedemstat do over the long term? We know these patients are going to take their drugs for quite an extensive period of time. So we have an extension study where patients who are willing are able to roll off any of these studies that we have onto the extension study to continue receiving drug and continue monitoring, obviously at a less frequent than what you would have on the primary studies, but we want to be able to see what's happening over time with bomedemstat patients. We also want to be able to see if we're getting any insights into, is it potentially disease modifying? Are the reductions in the VAF, the reductions in bone marrow fibrosis, reductions changing from homozygous to heterozygous, having an impact on the course of the disease? And we can only do that by following patients for a longer period of time than what you would typically have for phase 2 or phase 3 studies. So we do have that study open to continue to follow bomedemstat patients. Thank you for listening.
Thank you so much. It's so interesting. You know, I watch these drugs from start to finish, and then post, you know, after everything has been, you know, now they're looking at other things and new clinical trials. It's fascinating. Thank you very much. And lastly, but certainly not least, from Silence Therapeutics, Dr. Curtis Rambaran, who's the Chief Medical Officer there. How are you today, Curtis?
Great. Just get my slides up to share.
They look good. Oops.
It on. This should be better, I hope.
Yes, that's
Okay,
good.
great. So thanks again and for the opportunity to share the work that we are doing at Silence. You've heard a number of great presentations this morning and the breadth of studies in hematology, but certainly with the advancements in genomics and genetics, we have now a great opportunity to potentially pursue targets once considered to be undruggable, so not reachable by small molecules or peptides or other modalities. And I wanted to share with you today some of the work we are doing at Silence, and I'm the Chief Medical Officer. So Silence has got a global network. We have got headquarters in London. We have our molecular designers and preclinical scientists in Germany, in Berlin, and a lot of the senior leadership based in New Jersey, in Hoboken. So the name Silence derives really from silencing diseases, or gene silencing really. And as I mentioned, using the current advancement in genomics and genetics via small interfering RNA technology. So we have precisely designed sRNA molecules. So basically, this is matched to a target gene. So identify a target gene that results in transcription and then translation via messenger RNA of abnormal protein of interest. And we're able to design the sRNA to specifically target the messenger RNA responsible for translation of that abnormal protein. We have learned, a lot of ways along the way to optimize certainly the molecules. We also use proprietary linkers that connect with the sRNA molecule, and we use specific ligand. In the case of the hematology asset, we use a GalNAc ligand. This is a naturally occurring sugar, and it allows us to specifically target the liver. So the modality really is made up of a specific sRNA, proprietary linker, and then a ligand that takes the sRNA to the hepatocytes. So very much I show here, this is injected by subcutaneous injection. Because of the addition of GalNAc, it goes directly to the hepatocytes. It gets internalized via the ASGPR receptors. It enters the endosome, and then a portion of the drug gets out of the endosome, and then this binds to your messenger RNA of interest in the RISC complex, which is a sort of a multi-protein evolutionary complex that's really designed to degrade viral RNA, to protect you from viral infections. So we have now been able to use this machinery to target specific diseases, and to do it in a specific and a very targeted fashion by conjugation to the liver. As I mentioned, the name Silence is derived from silencing gene expression. So we are not altering the gene, and that's going to make that very clear. We are targeting the downstream, the messenger RNA, the sort of the signals that then results in translation into the abnormal protein. We are targeting both rare and common genetic disorders. The beauty about it is that it's durable. We have shown that in other programs also you can get long-lasting effects with single injections, but it's reversible. So once you stop treatment, you will reverse any changes. So here's a bit about our clinical programs. I'll just briefly tell you about zerlasiran. This is targeting lipoprotein A lowering in cardiovascular diseases. It's our most advanced candidate, now in late phase 2, preparing for phase 3. And the important thing to highlight is we have been able to lower lipoprotein A levels, an abnormal protein that's deleterious to the vasculature, around 90%. So we have been able to knock the expression 90%, and even after 2 doses down to 201 days, you still have very, very low levels. So some insight into the potential potency of the modalities. So I want to talk to you a bit about divesiran. This is our sRNA that targets TMPRSS6. You heard a bit earlier on some very nice work going on with hepcidin, the master regulator of iron flux. We are not given hepcidin exogenously, as you saw before. We are really targeting the endogenous pathways to sort of increase hepcidin, but in a controlled endogenous fashion. We have developed positive preclinical data showing potential utility, not only in polycythemia vera, but other conditions. And we have already demonstrated a good proof of mechanism in healthy volunteers. So our first study in healthy volunteers, we targeted 24 subjects. We showed an average rise in hepcidin fourfold and a 50% reduction on average of serum iron. So exactly what you would want to do, targeting this mechanism. And after a single dose, these effects persisted for at least 2 months. So it gives you some insight into the potential durability. Importantly, it was very well tolerated. We had no SAEs and we had no injection site reactions of any significance. And that data certainly supported us partly going into a phase 1/2 study in polycythemia vera. We presented this data a couple of years ago at ASH. So you heard a lot about polycythemia vera 2 presentations ago, so I wouldn't go over any more detail, but importantly, the objective is to target the hematocrit down to less than 45%, a good index of red blood cell accumulation, and to do that, in a fashion that is helpful to the patient, knowing that a lot of the PV patients are iron deficient at diagnosis, and this gets exacerbated by recurrent phlebotomies, which is sort of the current standard of care for most of these patients. So we have some very good preclinical data with divesiran, showing that we can, in the animal studies, we can elevate hepcidin, we can reduce red blood cell count, and do it in a very safe fashion, and this is published. We do have fast track designation for polycythemia vera and orphan drug designation from the regulatory authorities. What this really means in practice, it allows us very close collaboration, very frequent collaboration, and also an opportunity to be innovative, to try different approaches to study design and to get approval very early from the regulators. So our hypothesis really is that divesiran will elevate hepcidin in a very controlled fashion within the internal milieu, the internal physiological compartments of the body, and really control iron release into the circulation. So the objective is to move iron availability away from the bone marrow, because that's where you need it to produce red blood cells, and move it to other parts of the systemic circulation or organs or macrophages where it will not have a deleterious effect. We have the phase 1/2 study in polycythemia vera ongoing. This is targeting adult patients. Our objective is to look at safety and tolerability and pharmacokinetics and pharmacodynamics. So what is the systemic concentrations and how it affects our responses. We have our phase 1 study ongoing. This is an open label dose finding study. So we are currently able to look at the data in stream, and it looks very, very promising to date. We anticipate to have that into the public domain later this year. And we are now gearing up for a phase 2 study, which will be a randomized placebo-controlled double-blind study in adult PV patients. The primary outcome measure in phase 1 is really safety and tolerability and an early insight on the number of phlebotomies. For the phase 2, we'll be looking at subjects who are phlebotomy-free after treatment. So phase 1 ongoing, almost completed. Phase 2, we are currently preparing very aggressively. So just to summarize, we have shown that divesiran is safe and tolerable in healthy volunteers, and we also, I forgot to mention, generated some data in patients with thalassemia. It looks very good in terms of safety and tolerability. We have a long duration of action from a single dose. So this is where we feel we could contribute to the field. We can give it potentially to patients another alternative, potentially a modality that you can dose infrequently. It could be every 2 months, every 3 months. We have to certainly gather the data. Our ongoing trial called Senrico is designed to really identify the dose-related development. And we believe that the PV patients will benefit from a reduction of phlebotomies and a better control of the iron stores and a better redistribution of the iron away from the bone marrow and more uniformly, which we believe will translate into better symptoms, particularly a reduction in fatigue. The Senrico trial is currently has been predominantly ex-US to the rest of the world, but we are now open for recruitment in the US at a number of top academic centers, and we anticipate to certainly ramp that up into the near future. So I listed some sites here for additional information, both at clinical trials and internally at Silence. We are very excited about the potential of this modality, particularly its precise nature and potentially its very safe nature. And we'll certainly keep you informed, and we hope to continue contributing to the field and all of the work that certainly the MPN advocacy group is doing at present. So
Thank you so very much. Thank you very much. This has been fascinating for the last 2 days. And this was recorded, and the recordings will be going out later on this afternoon. Thank you so very much, and stay tuned for more webinars coming up in the near future.